So welcome back everyone from our break, and it's my pleasure to introduce our keynote speaker today, H Postlethwait from the University of Oregon. And our schedules for this conference, being on these kind of accordion flyers, don't give us enough room to print the abstracts. But if you take a look at John's abstract, he has probably the best first line ever for an abstract for a talk, which is fish know lots about water. And so we're going to learn lots about fish today too, and about tools for fish. John did pioneering work first with Drosophila and then getting into the fish world with zebrafish in developmental genetics, especially as it relates to sex determination. And he created the first genetic maps for zebrafish, which then anchored the zebrafish genome, became very important in all of the tools that we have now for zebrafish based on that incredible genome. A lot of important work on the evolution of development, and also pioneering work on genome duplications, which he's going to talk about today. And you'll probably learn about things such as paralogs and other kinds of logs as we go through today's talk. John also mentored me as I transitioned about 15 years ago from NSF funding to NIH funding. It turns out it's a pretty steep learning curve to make that shift, and it's really helpful for me to have a mentor during that time. And we've worked together all over the world, everywhere from the Bering Sea to Groote Island off the north coast of Australia. In fact, we're going to be out together in the Bering Sea next month on St. Lawrence Island. And it's just been a real pleasure to work with John over the years. Probably the most important thing I should tell you about John is his running. So John runs marathons, and he had and much longer distances. And he had run a marathon on every continent except for Antarctica. So he organized a marathon in Antarctica and ran it in snowshoes in order to get that achievement. And John also, for his birthday every year, would run his age in miles. And I think it was when you hit early 70s, you switched to kilometers, as I recall. But
no, it's 70 miles.
You got to 70 miles.
70th
Okay.
Then I switched to kilometers.
And 71 switched to kilometers. So that was a pretty good switch. And actually, we're doing a nice long run together tomorrow. So anyway, with that, we'd like to have John come on up and deliver our keynote, integrating human, animal, and environmental health in the water, fish as models. You may have to turn on your mic.
Yeah. Thanks, Frank. And it's good to be here. I've enjoyed the talk so far, and it's been great working with Frank in these nice places over the years, too. So if I can remember how to use this thing and advance the slides. So topic is water, and of course fish do know a lot about water because they've been doing immersion learning about this for a long time. They're good for reporting about environmental health, and they're good for reporting about making models for human health. So those are basically the two things that I'm going to talk about. And so we're going to talk about these topics: genomic similarity of fish to humans or other vertebrates, similarity of developmental mechanisms, of cell functions, how easy it is to look at for mechanistic studies in fish to learn about what causes things to happen, and then thinking about evolutionary. For these kind of things, we make laboratory-induced mutants, but it's very interesting to look at evolutionary mutants that have happened naturally, that also are good disease models, and then thinking about biomarkers for healthy waterways. And if Sophia doesn't get after me too badly, I'm going to add at the end something about global fish health and global climate change, which I didn't put on the outline there. So this first section may be a bit dense. So we're thinking about genomics and comparing the genomes of humans to fish, to zebrafish particularly here. So fish have great genomic similarity to humans, or other vertebrates. And we can see this if we look at most genes on zebrafish chromosome 3, which is DRE3, have orthologs, that is, copies derived by descent from a single copy in the last common ancestor, on human chromosome 17, Homo sapiens chromosome 17. So this represents the length of human chromosome 17, the length of Danio zebrafish chromosome 3, human chromosome 17, and comparing, what I'm going to show you is comparing genes on those two chromosomes across the chromosome. And what we have here then is here the zebrafish chromosome 3, human chromosome 17, and each line is connecting orthologs, identical copies of the genes in the two different species. And what you can see is that most of the genes, nearly all of the genes, along the zebrafish chromosome have corresponding genes in human. The orders are different, but the content, the chromosome content, is very similar. This is work that One Health did in the lab. The differences that you see are mostly by inversions, where a chromosome would break and then would invert so that you have a little inversions. And those happen very, very frequently during evolution. So much of the chromosome structure is preserved for 350 million years, a long time. It's amazing, since the human and zebrafish divergence. The zebrafish copies, the orthologs of about 80% of the human genes are conserved in zebrafish. But despite this similarity, it's strange to learn that zebrafish chromosome 12 also has orthologs from human chromosome 17. So if we look at just same kind of thing, you see this very same kind of thing with zebrafish chromosome 12. And then if we compare, look at the two together, what you see is this is human chromosome 17 here, human chromosome 17 here. Both of them, throughout the whole length of both zebrafish chromosomes and the human chromosome, are sharing their genomics. So what's up? So how can we understand this? It's, what this represents is double conserved synteny. So synteny means two genes are on the same chromosome, and it's conserved when humans have a pair of genes on a chromosome and zebrafish has a pair of genes on a chromosome. So that's a conserved synteny, conserved for 350 million years. The way we can understand this is this. So again, here's human chromosome 17. Well, let's see. If I go back and look at this again, and it just blow up. I'm just now taking this region and blowing it up to here. And so that you can see each box here represents a gene. And what we see here then is that in this illegible red, you see that some genes on human chromosome 17 have two copies in zebrafish, one on 12, one on 3. Some genes on human chromosome 17 have a copy only on zebrafish chromosome 12, and some have a copy only on human copy on 17 and a copy only on zebrafish chromosome 3. So how can we understand that? And this is a general thing. This is the same for all over the entire zebrafish chromosome, genome. That's the same thing. So we can understand that if we assume here, again, each little box is a gene and we have a chromosome with genes on it. And then there's a duplication. So immediately after the duplication event, there were two chromosomes that looked like that. And then over time, evolutionary time, what happened is that some of the genes disappeared from one chromosome or the other. Generally not both. So that left us with some genes on both of those duplicate chromosomes, some genes on one, some genes on the other. And as a generality, about 20% of the human genes remain in 2 copies in zebrafish, and in fact, most teleost fish, most fish that you're familiar with, this is true. So what did that, so we discovered that a number of years ago and wondered what's going on. So it turns out then that this can be understood, since it's across the whole genome, that can be understood only if there was a whole genome duplication in the fish lineage. And so then we can ask, when did that happen? In other words, there was an ancient fish that had 23 pairs of chromosomes, let's say like humans did, and then all of a sudden they had 46 chromosomes. And so when did this happen? So here we have fish phylogeny, starting with these lobe fin fishes like humans and chickens, and then some ancient lineages of fish like bowfin and gar. And when you look at these ancient lineages of fish, what you see is one gene in human to one gene in these fish. But when you look at teleost fish, you see generally one gene to 2, or at least one chromosome part in human to 2 chromosome parts in the fish. And so here we have the different, so these are teleost fish that have this 1 to 2, and basically this is 50,000 species of fish, and this is half of all vertebrates. Half of all vertebrate species have this relationship to the human genome, a genome of chickens and frogs and so on. So this event then is an ancient event. It's shared by all teleosts, and so that means that the event happened very early in teleost fish evolution. And so we call this the teleost genome duplication event. It happened after the divergence of gar and bowfin, and then before the divergence of all species, the eels, bony tongue fish, salmonids, goldfish, whatever, tunas, whatever fish you want to talk about. Or of those 50,000 fish, anyway. Okay, so then, in summary then, there's great genomic similarity, but it's modified by this genome duplication. Zebrafish has about 80% human genes. Genes along the chromosome tend to be conserved, and you had this teleost genome duplication event. Not only are genomes conserved, but also there's developmental mechanisms are conserved. So here I want to, oops, my finger's a little bit, whoa, how far did I go back?
We want to see it again. It's good.
Yeah, right. Sorry about that. I'd kind of like to start that movie. I didn't make it stop. So Don Kane made this movie, a postdoc in the lab. This is the yolk of the fish. These are cells on top of the yolk. They're dividing. You can see them divide simultaneously initially. Then you see the fish start to gastrulate, comes down. Some of the cells go inside, comes around the yolk. Right here, as the head, the eye appears. You see the precursors of the vertebrae appear. And in 19 hours, you get a little baby fish, and in a zebrafish embryo, you can look at this stage and later stages too, and you can see every cell. Well, you can see some of the cells. That's the size of cells. You can see each of those bumps there is a single cell, and you can see those throughout the entire embryo, which is a huge advantage when you're doing mechanistic studies. And this is very similar to the human embryo. And you can just look down and you can see, looking at the hindbrain, this very thin roof of the hindbrain here, very thin roof of the hindbrain there in the zebrafish. You can look and see the ear of the zebrafish, the ear, dark oval there in the human embryo. This is a 28-day human embryo. The gill arches, you can't see the gills in the fish very well, but you can see the gills very well in the human embryo. Looking down, look at the eye in both of them. The heart is easy to see. The heart beating in the zebrafish is easy to see. The fin or the limb, large here in the human embryo, and it's right here. You can see it in the zebrafish. Look at the notochord, this tube here, cellular tube here that you can see in both. And then you can easily see the vertebrate primordia here in both. So, and of course then the tail of the human and the tail of the zebrafish. So the early embryology of the human and the fish are very, very similar, but the fish embryo has a yolk right here, and the human has the umbilical cord, so there's some differences with respect to that. But otherwise, the organs are forming just the same. The pancreas, the liver, the gallbladder, all of those things are forming just the same in fish and in humans. Fish don't have a prostate and they don't have lungs, but other than that, they're very, very similar. So what is the consequence of that? How can we put together that idea that there was this genome duplication event and the idea that the developmental mechanisms are similar and the genes are similar? So Alan Force was a graduate student in the lab, and we're trying to wrestle through this, and he's a very smart guy, and these are his thoughts. So let's think of an ancestral gene where it had a coding part that's coding for protein and regulatory elements that regulate where and when that gene will be expressed. After duplication, then everything about the gene is going to be duplicated. It's regulatory elements and its protein coding parts. And then Alan had the idea of that he called subfunctionalization. And this is this. To preserve both copies of the gene, both copies have to be essential for development. Because if a gene isn't essential, it's lost. If you don't use it, you lose it, right? So both copies then have to be essential if they're going to be saved for 350, well, for 250 million years since the event. So he said probably what happens is you get reciprocal loss of regulatory elements or reciprocal loss of protein coding parts, which I didn't show here. And so now then both copies are positively selected. This one has to be saved for evolution because it is necessary for whatever this element causes, but it can't do this function. This element is necessary because it does this function here, and this gene is necessary because it does this function and it can't do that function. So you have to keep both of those. So that's the idea of subfunctionalization. In addition, you can have preservation of duplicate genes by neofunctionalization. And for neofunctionalization, you can have the origin of a novel essential function, something that's positively selected that evolution likes. In order for this to work, you've got to have the loss of an ancestral function, essential function. And so when that happens, then you can have evolutionary novelties occur. That's in this paper here. So what does this mean for development? So if we look at a mouse embryo, engrailed gene, engrailed 1 in a mouse, it's expressed in the isthmus of the brain. It's expressed in specific hindbrain interneurons. It's expressed in the limb bud, just here, the part of the limb bud up here, which grows out and causes the limb to grow. So just these two thin lines on the forelimb and the hind limb of the mouse. It's expressed in the spinal cord. So in a zebrafish has two copies of engrailed 1A and engrailed 1B. And the engrailed 1A gene is expressed in the isthmus. You're looking at a fish at its back of the fish, and this is this part of the fish. And it's expressed then in the isthmus, in Grail 1A is. It's also expressed in the limb bud. So down here, well, here on the shoulders of the fish, you can see the expression there, and it's expressed in the spinal cord. The other copy in Grail 1B, it's also expressed in the isthmus. It's expressed more strongly in the isthmus. It's expressed in these hindbrain interneurons, where the in Grail 1A gene is not expressed, but the mouse gene is expressed. And then Grail 1B gene is not expressed in the limb bud, but the mouse gene is expressed in the limb bud. So you see what you have is this situation where you have the ancestral gene, in Grail gene, was expressed both in the hindbrain interneurons and in the limb bud, but after this subfunctionalization event, those functions sorted out to the two different genes. So to understand fish development, what we have to do is to understand what's happening with that 20% of the genes that are retained, 25% of the genes that are retained in duplicate. This is what's happening to keep those genes around. So we've got genomic similarity, we've got developmental mechanism similarity, we've also got similarity of cell functions. So in the last few years, it's been possible to interrogate the transcriptome, that is all of the genes that are expressed in single individual cells. And so you can now take any organ you want, a liver or brain or whatever, here the testis, and you can dissociate the testis into single individual cells, where each dot there is a cell of several different types, cell types. You isolate those cells, and then you sequence the messenger RNAs in each cell separately and keep them separate, and then you can identify what genes each individual cell is expressing. And so then you can then do bioinformatics and compare each cell in a multidimensional analysis, compare the genes that are expressed in each cell to the genes that are expressed in each of the tens of thousands of other cells in your experiment. And then plot the cells that have similar gene expression patterns. You plot them close together, and ones that have different expression patterns, you plot them far apart on this basically a principal components plot. So this is these data are from this paper here, and this is a human testis, human embryonic testis. It's a fetal testis. And what you see then is groups of cells that have similar expression patterns in each of these clusters. And you can then, because you know what some of the genes that each cell type is expressing, you can label these as stem cells or differentiating spermatotids or spermatocytes or Sertoli cells or whatever. You can identify which each is. And so Catherine took in the lab, took testes from zebrafish, wild type, and did the same analysis. And again, each dot here is a single cell representing the transcriptome of a single cell. And then we can compare the zebrafish to the human. And we can look and see that these stem cells in the human, look to see what genes they're expressing, compare it to these different clusters in zebrafish, and see that these clusters down here then are the stem cells. And we can look at then the differentiating cells, and we can see that those are the differentiating cells. And so then we can look at the other cell types of the sperm, and you can see that there's a kind of an arc of going from early stages to late stages here, and we have that same arc in zebrafish. And so the gene expression patterns and the cell types are very, very similar as defined by their gene activities. And in addition to the germ cells, these other cell types of the gonad, the same thing. So there is, at the single cell level, there's also greatly similarity. And then we looked at the zebrafish mutant for BRCA2. So BRCA2 is the second most common gene that's mutated in hereditary breast cancer. And we made a mutation in that gene in zebrafish. And then we can compare the wild type zebrafish testis to the mutant. And I'm not sure if you can see that, but you can see that these forms that have gene expression very similar to the late stages in the human, they're missing from the mutant. So we can then see that the mutant, that this gene, BRCA2, is essential then for sperm development in zebrafish. So we're doing a lot of these kinds of single cell studies to try to compare human and zebrafish cell types. So even at the single cell level, human and fish are very, very similar. But in addition then, it's easy to do mechanistic experiments in fish. So the NIH has this program called the Undiagnosed Diseases Network. So in this NIH program, people that have diseases, generally children, that there are a large number of children that have diseases that can't be diagnosed. They have a series of pathologies, and they're not recognizable. They're specific to this child, generally quite rare and undiagnosable. And it's quite a problem for the families and of course for the person, the child as well. And so at the NIH then, the affected person and generally their parents come and get a very stereotypic and very in-depth clinical analysis of what's of their pathology, and they get either whole exome sequencing or whole genome sequencing to learn what the protein sequences are, or the whole genome sequence is across all of the proteins, all of the genome. And then frequently they come up with a gene that nobody studied before. And so we don't know if that gene actually has an effect on physiology or what that effect might be, because nobody has studied it. And so NIH then sends to our model organism screening center the sequences and the phenotypes, and then we look at them either in Drosophila or in C. elegans or in zebrafish. Now there are other labs, lab at Baylor and a lab at Washington University that do the invertebrates, and then we do zebrafish in our lab. And so then what we do then is we make the zebrafish mutant in the ortholog of the human gene, and then we ask, is it reasonable, does the fish have the same disease as the person? Okay, and if they do, then that validates the assumption that this gene is causing this person's pathologies. So in this case, UDN number whatever, the person presented at age of 16 years. He had primary hypogonadism, that is, his gonads never grew. They were always small. It wasn't a matter that they grew and then regressed. They were just never grew. He had azoospermia. He didn't make sperm. He had low testosterone levels. Had 46 chromosomes. Had 2 X chromosomes. He did not have a Y chromosome. and he did not have this is the SRY is the gene on the Y, the only gene on the Y that's essential to make a testis comparing to an ovary. And he had a mutation here in a gene called NR5A1. And it changed a specific amino acid here. So the question then is this mutation responsible for this person's clinical features? And so Eileen in the lab made a mutation in, so there are two copies. It shouldn't surprise you now that there are two copies of this gene in zebrafish. And so Eileen knocked out both of these genes using CRISPR technology. And the human mutation is here. This amino acid, R amino acid here, is widely conserved across all vertebrates in this protein. And it was changed to a W in the human, in the patient. And then Eileen made mutations very near to where that mutation is. And then we want to see if that has an effect that would be similar to this person's pathologies. So in a wild-type fish at 35 days post-fertilization, some of the fish have gonads that have oocytes in them. And so those are fish that are making ovaries, and they're going to become females. Some of the fish have gonads that have young spermatocytes in them. So those are making a testis, and those fish are going to become males. In the mutants, the NR5A1A mutant, the gonads didn't grow very much. Some of the gonads made some oocytes that then died, but many of the gonads didn't make any oocytes. And then in the NR5A1B mutant, none of them actually made oocytes, and the gonads were quite small. They never really differentiated. They were undifferentiated gonads. And in the double mutant, where both genes are knocked out in the same individual, the gonads were even smaller, just this little bit right here, this little bit right here. And so this then, the zebrafish mutants have this primary hypogonadism. They're very, very small. They have the azoospermia. They never made sperm. So they have these, they mimic these in the patient. And then Danielle Dillon and Lauren Buck's lab at NAU measured the testosterone levels in the mutants and in the wild type. So this is the wild type male. This is a high level, and this is a log plot. So these are not orthologs. These are real logs, your kind of logs. And so this is the male with very high levels of testosterone. The female has low but measurable levels of testosterone. And here are the three mutant types, the single mutants, the two single mutants, and then the double mutant. And they all have lower testosterone levels than even the female. So then the patient had low testosterone. The zebrafish mutants have low testosterone. And so this then is a good model, and we conclude then that zebrafish mutations cause phenotypes like the undiagnosed disease network case. Supports that this gene is broken in the patient. So zebrafish are good ways to study, make mechanistic investigations, and so there we were using mutations that we make in the lab, but what's very interesting is mutations that are made in nature. So Charles Darwin said, "I'm only surprised that more wrecks of ancient life have not been preserved." So by wrecks of ancient life, he meant cases in which there are species that have lost ancestral essential features. And so they give an evolved phenotype that would be pathogenic in the ancestor. So one example that he used was were cave crayfish that have eye stalks but the eye is gone from the top of the eye stalk. So the cave fish, the cave crayfish has no need of the eyes. There's no light, and it lost the eyes, but it kept the stalk. So he called these wrecks of ancient life. So these then are quite interesting, and here Fenway Mung, postdoc in the lab, compared cave fish, these Cynocaechalus cave fish, to the surface close relatives. And what you can see here is the pigments are different, and the cave fish has a small eye right here. You can see this little darkness, but it's very, very small, and it never gets out of the, never comes to the surface. So it can't form images. And so then Fenway looked at the brain, and he saw that this part of the brain that is involved with vision is much smaller than normal. And then he did transcriptomics, and he found that the brain transcriptome had lower amounts, lower gene expression from these genes. Well, all of these genes are involved in energy metabolism, and so the cave environment is altering the whole energy metabolism of the cave fish. And it turns out, looking at other cave fish, here Nick Rohner paper here, has shown that these fish, basically they have diabetes. They have severe diabetes, and they the important point here is that looking at how these cave fish deal with their diabetes may provide some insights into what we could do for people who have diabetes, therapies for diabetes. In another example I want to give is blackfin icefish. So the icefish lives in Antarctica, and these icefish are the only fish, the only vertebrate of any type that lives without any mature red blood cells and without any hemoglobin genes. So the fish is white because it has no hemoglobin genes. And here I show three fish, two ice fish, different species of ice fish, and a closely related red-blooded fish. And this is looking at the heart. If we look at the heart is red, partly due to the blood, but mostly due to the myoglobin protein that's in the heart. This ice fish has myoglobin in its heart. It has a pink heart. This ice fish has no hemoglobin or myoglobin, so its heart is not even pink. And then if we look at the blood, this fish has red blood. It has hemoglobin genes. This fish has myoglobin, but it has no hemoglobin, and it has no mature red blood cells. And this fish has neither myoglobin nor hemoglobin, and it has no mature blood cells. They have very large hearts, and their blood vessels are very, very large. They get some oxygen across their skin, and yet it must be detrimental not to have the red blood cells and the hemoglobin because their heart is enlarged to take care of that. So I wondered which was lost first, the red blood cells or the hemoglobin. Both are gone. Which was lost first? And so Thomas, postdoc in the lab, and Zoe, an undergrad in the lab, looked at the blood cell morphology in all of these species of Antarctic fish. And so here's a normal, the red-blooded fish, that fish, and stickleback and all other kind of fish. They have oval red blood cells that are flat, but they have nucleus. So they have nucleated red blood cells that are different from ours, of course. These are red blood cells from a plunder fish. So plunder fish on this phylogenetic tree are here. And then these are from a dragonfish, and the dragonfish on this tree are here. and then the ice fish on the tree are down here. And this measures the area of the red blood cell. Red is small red blood cells. Blue is large red blood cells. And what you see is, first of all, that the ice fish have the few red blood cells that they have. They're immature, but they're very, very large. And that trend from small blood cells to larger blood cells started in dragonfish before the evolution of ice fish. And then they also looked at the roundness of the red blood cell, whether it's oval. The zebrafish and other fish have oval red blood cells, but here dragonfish and the few cells that they could find in the ice fish, the immature cells, were also very round. And that trend also started then in the before the ice fish. So that trend started here. So what I conclude then is that it looks like the red blood cell loss happened before the loss of the hemoglobin molecules. And so it's interesting then that when the sequence of dragonfish was determined, that the dragonfish then has mutations in the dragonfish gene that in humans, when it's mutated, it causes the disease spherocytosis, severe hematological disease. So, so there are these evolutionary mutant disease models. They have phenotypes, some that mimic human diseases like this profound anemia of the ice fish. And learning how they cope might help to suggest therapies. And then thinking about biomarkers then for healthy waterways. So Frank has been studying this problem in Alaska and other places for many years. So the problem is the persistent organic pollutants like polychlorinated biphenyls, which people from the Arctic, from the poles, are especially exposed to at high levels. And so here on Sivakak, which is closer to Russia than it is to mainland Alaska, people live, and they're living near formerly used defense sites, which provide a point source for these pollutants. And remember when back in the Cold War, when we had over the horizon radar because we needed to check up on the Russians, because back then we thought the Russians were bad people, but the Cold War is over now and we know that that's no longer true. So we tore down those radar sites because Russians no longer like to invade other places, and they provide now a point source for pollutants. Another way that we get pollutants in the north is from global distillation, where pollutants produced down here then rise and then fall as in precipitation in higher latitudes. So Lauren Buck from NAU and Frank and I and others went out to look to see what's happening on Sivakak and wonder what can fish tell us then about the effects of PCBs on living things living on the island. And so we collected from a number of sites going up the river here samples of fish and water, and then looked at the PCBs in the fish in either stickleback fish or Alaska blackfish. And here in these sites here, this is where the old defense site was. These sites are above where the water flows. And so they are less polluted from this point source. They would still have pollutants from the global distillation. But they have low levels of PCBs in the fish that we caught there. In the fish that we caught here in these sites directly downstream from the site, from the defense site, they had very high levels of PCBs in them. And that had consequences. And the consequences they had was, I'll show you in a minute. But we wanted to look at a site that was well away from the point source. And so Lauren and I hiked out here to the Tappy River and collected and dissected stickleback fish there. We needed a flat surface to dissect the fish. The only thing we could find there to dissect on were whale vertebrae that were there on the shore, and we used that as our lab bench. And so analysis of those fish then, here is the yolk proteins that are made in males. Now, males don't normally make yolk proteins. Females make yolk proteins. They're caused by estrogen. And so those fish at the Tappy site out here had very low levels of yolk protein. But when we looked at, and then when we looked at the upstream site, also there was some yolk protein, but not an enormous amount. And then when we looked at these downstream sites here, what we found was large amounts, some variability, but large amounts of yolk protein. And so the conclusion there is that these pollutants are then having an estrogenic effect on the males. And so we wondered then, is that having an effect on gene expression? And so we took the fish and cut their tails off, cut their heads off, and took the center, which has their liver and their gonads, and we did transcriptomics on it, looking at the messenger RNAs present. And what happened was then, and this is again is a principal components analysis plot, and the ones that were slightly contaminated separated from the ones that were strongly contaminated quite well, so there's quite an effect. And then when we looked at did an analysis of what genes are affected, what we found was that genes were downregulated, genes in the contaminated, more heavily contaminated fish, that a bunch of genes that control cell cycle and cell division were downregulated. Genes that affect DNA replication and mitosis were downregulated. Genes that are involved with DNA damage and DNA repair, most of those were downregulated. So we conclude that stickleback from these contaminated sites have decreased capacity to respond to cellular damage by replication and DNA repair. This work was also done by my lab manager, Tom Titus. So then the question is, what will happen to the people? What's happening? Are these things also happening to the people in Sivakak, to the indigenous people that live there? And so that's biomarkers for a healthy waterway. And if I have, so I'm going to take just two minutes to just do one last thing about fish health and climate change. So we were fishing in Enverd Bay in Antarctica, and here a chart for temperature change, and Enverd Bay is right here in Antarctica, and with postdoc Thomas Devine, and we found fish that have had these tumors. About 30% of the fish in this catch had these tumors. We had never, in years of fishing in Antarctica, we'd never seen fish with these tumors before. And now in the one tow, we had really hundreds of fish that were affected. They had these very ugly tumors on various parts of their body, often quite substantial parts of their body. And so we wondered, what is going on here? What are these? And so we did collected samples for histology, and then you see these dark purple cells are the host cells, and these cells here, there's a cell, those are the cells that don't belong there. Those are cells that are causing the disease. And so Thomas took the DNA that I had collected, and he sequenced it, and he found that the DNA that was similar to ciliates, the protists that are ciliates, and like paramecium, related to paramecium, apicomplexa, and there had been a recent paper that showed a similar kind of thing, or a species that came out in the same phylogenetic portion of the tree, and they called this X-cell disease. So this X-cell disease then, so these cells are parasites. They're a protist that's causing this, causing these neoplasias or xenomas. And so then Thomas made probes for the fish cells. So now this is a cross-section of part of the skin, and where there's blue here is an in situ hybridization, which shows you where the fish cells are in this preparation. And this is then the adjacent section, and he stained it for in situ hybridization for the X cells. And what you see is a reciprocal where there's blue here, there's white here, where there's blue here, there's white here. So these then are the X cells, and so there are compartments made by the host that are occupying or surrounding the xenoma cells, the ones from the parasite. And furthermore then, Thomas looked at the weight of the affected fish and our controls that were unaffected, and there's substantial difference in weight. And so we conclude that this weakens the fish, or that these fish are unhealthy, that the disease causes unhealthy fish. And so so there's substantial climate change happening there, and we wonder if that change is either strengthening the parasite or maybe it's weakening the fish and makes the fish more susceptible to the disease. So that's it, and there's the outline. I won't go over it again, and just we'll take your questions. Thank my colleagues at Oregon and my colleagues in Arizona, and thank you.
Thank you, John. Fish questions.
I'll ask the questions. There's a little bit more hesitancy. What other plausible explanations are there for this big change in the parasites? As in, have there been other huge changes in that region besides climate change that someone could blame it on?
So nobody has seen this kind of outbreak in Antarctic fish before, or in any fish before, really. And so we don't know what is changing. So there are two basic things that are changing in the water in Antarctica. One is the temperature. Now, the temperature hasn't changed very much, actually, yet. But it's the air temperature has changed a lot, and the weather is changing. The temperature of the water has increased only slightly. The second thing that's changing is the salinity because the ice sheets and the glaciers are melting, the water is becoming less salty. And so it may be that that is encouraging the dispersal of the parasite, or it could be that it makes the parasite, or it could make the parasite for some reason more easy to infect the fish, or it could be that it affects the fish so that the fish is under stress, and that it makes the fish less able to keep the parasite out. And I don't know which it is.
Do we know if there's significant chemical changes in the water due to the change in the temperature difference between the water and the air?
Mm-hmm. Okay. Question is are there chemical changes in the water? Two things. One thing, the water's cold there. I've swum in it twice, and it's cold. It's minus 1.5 Celsius. It's 28 degrees Fahrenheit. And cold water like that holds more oxygen. And so these ice fish that don't have any hemoglobin or red cells, mature red cells, so they are very susceptible to oxygen stress. And as the temperature goes up, there's less and less oxygen. And so that's one chemical change that happens in the water. The second thing is, though, there aren't a lot of pollutants in Antarctica. The polar circumpolar current provides a barrier between the water that's in the Atlantic and the Pacific and the Indian Ocean to the Southern Ocean, the Antarctic Ocean. And so there's not an exchange, and so there are not really many pollutants at all in the Antarctic Ocean.
I would think, though, that with global distillation, you would have a good deal of air pollutants, and that if you have a large change in temperature in the air, then that would change how much of those are entering the water.
I would think so too, but I don't know if that's true or not. So she's thinking about global distillation and whether changes in the temperature would change the pattern of global distillation, which would bring pollutants to the Antarctic. I don't know.
John, I have two questions for you. One is, did you swim before or after the marathon? And then secondly, you had alligator guard in your lab, and you sequenced their genome. Can you talk about then making a comparison between the human alligator gar versus a human to a teleus? So for disease genes.
So we sequenced the gar, so I'm not sure I understand the question exactly, Frank. But the weird thing is that the gar genome is very, very similar to the human genome. As a matter of fact, to tetrapod genomes. So the human genome is rearranged some compared to turtle genomes and chicken genomes. Chicken genome is very conserved. Chicken genome and gar genome, most of the chromosomes in chicken have a single chromosome that's identical in gar, which is really, really quite a weird, totally unexpected thing. So there's been quite a conservation there, and zebrafish has had a lot more changes than gar, and then humans have had a lot more changes than chickens. And with respect to the swimming question, I swam after.
Other questions for John? Thank you. We'll move on to our panel.